So the story I'm going to tell you today begins with the topic of collective cellular migration. So here's an example of collective cellular migration. These are MDCK cells advancing over a cell-free region, and you can see how the cells migrate to close the void. This is an example of wound healing. So collective cellular migration, let's get this out of the way. So collective cellular migration is important for wound repair. This is a model of wound repair, embryogenesis and morphogenesis, growth, regeneration, remodeling, cancer cell invasion, metastasis. None of these things can happen without collective cellular migration. So collective cellular migration is pretty important in multiple dimensions of biology. So this has been recognized for a long time. So the question is, what are the physical forces that are involved? Because if these cells are moving, there must be physical forces that are moving them along. And the question is, what is their nature? So here's kind of a cartoon diagram showing a kind of a cross-section of an enhancing epithelial sheet. The red arrows represent what are called traction forces. Those are forces exerted by the cell on the substrate on which it's crawling, and the blue arrows represent intercellular forces, forces transmitted across cell-cell junctions. So the question for a long time is, what is the nature, the magnitude, and the distribution of these physical forces? And from 1914, that's the earliest reference I know to collective cellular migration, until 2009, that question, the nature, magnitude, and distribution of forces, had been pure guesswork. And some of the guesses were this. One is, it's all about proliferation. Cells proliferate, and they create a pressure that pushes the front forward. So that was one of the guesses. The second guess is it all has to do with these leader cells. I don't know if you can see my pointer, but these leader cells seem to have be specialized for crawling in terms of actomyosin content. So the idea is that these leader cells pull the front forward, kind of a locomotive pulling the train behind it. And yet a third guess was that it's like soldiers marching in a column. Each one is self-propelled. There are no forces between the soldiers. Each one is just crawling forward. So that's what was in the literature. It was in 2009 that actually this went from the realm of speculation into the realm of measurement. Jabi Tripat showed us in our lab, showed us how to actually measure these traction forces. And then a few years later, Dhananjay Tambe, also in our lab, showed how to measure these intercellular forces. And what we found stunned us, because it was nothing like anything we were thinking about. So here's what you see. So for this very epithelial layer that's crawling forward, these are the images of the traction forces that the cells are exerting on the substrate. So there's a color code here. Red indicates tractions that are pulling the front forward towards the void, and blue represent forces that are actually in the opposite direction, that are retarding the advancement. They're either cells that are being dragged along or even pulling in the wrong direction. And there were several stunning things here. One is you can see the leader cells are pulling, kind of almost uniformly pulling, but you see plenty of forces way behind the leader cell. You'll also see that there's plenty of cells that are pulling in the wrong direction. And then finally, the heterogeneity that's here, it's dynamic. The cells are flickering on and off, kind of like a Christmas tree. Cells are pulling in one direction one moment, a different direction at a different moment. So a given cell doesn't pull in the same direction all the time, and a given region isn't being pulled in the same region at the same time. So this was kind of a stunning development. Here's another way to look at the same data. This is looking at the traction force inside the circle here as the layer sweeps over that point. This might be my favorite slide ever. So what you can see is those forces are fluctuating like mad. It's not like in many systems we see maybe a mean value with small fluctuations on top of the mean. This is the opposite. This is huge fluctuations and a small mean value somewhere down here, around 10 maybe. So it's all about the fluctuations. So the take-home message from these findings were that tractions persist many ranks behind the leading edge, number one. Number two, the ranks behind, when you add up these forces in terms of how they're pulling the front forward, the ranks behind actually contribute far more than the leaders. The leaders are pulling, but they contribute a minuscule amount compared to the integral of all the cells behind. And the fluctuations are much bigger than the mean. The mean values are teeny, the fluctuations are huge. So what this is telling us is the collective migration is really kind of organized chaos. I mean, this is chaos. And some people call this dynamic heterogeneity as opposed to kind of structural heterogeneity. So in wound healing, in morphogenesis, in cancer invasion, but what accounts for this kind of behavior? Well, why does this happen? That was the next question. And I want to try to tell you the conclusions at the beginning in case they're not clear at the end. I'm going to try to convince you that this has to do with something called the unjamming transition, UJT for short. And we think it's fundamental to wound healing, morphogenesis, and cancer cell invasion. I'll explain in a bit what the unjamming transition is. The unjamming transition turns out to be complementary to, but should not be confused with the epithelial to mesenchymal transition, which is also associated with epithelial cell migration. It's distinct, and I'll show you that as well. So a few thoughts about this. When we entered the field of epithelial biology, a little over, well, 10 to 15 years ago, I was struck by certain tensions. One is that the epithelial layer, and here shown in the right is an airway, cartoon rendition of an airway epithelium. The epithelium can be solid-like, that is to say, each cell stays at a fixed spatial address, and if you perturb it, it springs back, so it's like a solid. Or it can be fluid-like and flow, and I showed you flowing with, like in wound healing or in growth. So in morphogenesis, growth, remodeling, wound healing, there's this tension between solid-like and fluid-like behavior. There's also a tension between precision, because on the one hand, the epithelium is responsible for mucus clearance, secretion, fluid balance, barrier and immune functions, and has to regulate all these very precisely. But also the layer has to be plastic, mechanically plastic, so that it can grow, adapt, heal wounds, and so on. There's another tension in terms of energy metabolism. You want to have an economical state, something that's not metabolizing a whole lot of ATP. But when the cells are migrating, that's energetically expensive. So there's this trade-off in terms of economy versus expense. And then finally, the question of order versus disorder. Many biologists think of an epithelium like this one in terms of a hexagonal grid, like a honeycomb with a regular hexagons. That's not what's going on here, and it's not what's going on with the migration. It's disordered. So you have this trade-off. I mean, I come from kind of engineering and physics, and there's this tension between the exactness of physics versus the messiness of cell biology, right? Everyone knows that biology is messy. And for someone from the physical sciences coming into biology, this is a humbling science to us because of all this noise or heterogeneity. So I want to say a few more words about disorder, because that's going to be a theme of what I'm going to talk about. So to biologists, the notion that the system is disordered is like, so we all know that. So is that just the nature of things? That's how biology is, it's disordered? Or could it be, and this is a case I'm going to make, could it be that disorder is an essential feature of multicellular life? Feature that's been overlooked, but essential. If you didn't have disorder, you couldn't have life as we know it. So I'm going to try to make that case as well. So who cares about epithelial unjamming anyway? So this brings me to the topic of dynamic heterogeneity. I've covered the first topic, life at the edge of chaos, now dynamic heterogeneity. So this is an airway epithelium, and you can see ciliated cells, goblet cells. It turns out the airway epithelium we now know is comprised of 12 distinct cell types, published recently or in the last year. 12 different cell types. Now, in terms of asthma, my lab has been focused on asthma for a lot of years. We know that a hallmark of asthma is aberrant airway remodeling, not just bronchospasm, but aberrant airway remodeling. And we know that in this remodeling, the airway epithelium plays a key role, as does viral infection. So this was about 10 or 12 years ago. I went to Jeff Drazen because Jeff Drazen was studying airway epithelium with Dan Chumperland and with Jinnah Park. And I said, Jeff, when you look at these cells in culture, do you see them migrating, or do you see some kind of strange migration phenotype? Because I said, I think we have reason to think there might be an aberrant migratory phenotype in asthma. And he said, Jeff, no one thinks about that. He said, yeah, there are a few labs that do scratch wound healing assays, but we don't think about these cells as being migratory. We think about these cells as being sedentary. They just sit there. They're like tiles to cover a floor where they do their barrier function. And as cell-cell junctions break down in disease or an injury, the grout between the tiles breaks down and the barrier becomes leaky. That's how we think about it. So that actually wasn't terribly helpful. So I went to Jinnah Park, who at that time was a trainee with Jeff. And I said, Jinnah, what do you know about migration of airway epithelium? And she said, well, no one's ever measured it, so why don't we do it? And so she did. And these were actually studies where Jen Mitchell played a big role. That's her picture in the lower right. So these are primary airway epithelial cells. These are primary cells. They're grown in air-liquid interface culture. They become well differentiated and pseudostratified. This is like the gold standard in vitro model of airway epithelium. So I'm going to show you the movies. On the left is the control, and the right is a stimulus I'll tell you about. So here's the control. This is a movie. May not look like a movie because nothing's moving. It's kind of like what Drazen said. The cells are just sitting there, each one in its own place. Now, on the right, I'm going to show you what happens when you mechanically compress these cells, apical to basal compression, which simulates certain aspects of bronchospasm. And here's what you see after compression. Same preparation other than compression. And you see a striking migratory phenotype. In addition, you can see interesting changes in cell shapes compared to the left. And it turns out you see the same type of response qualitatively, not just with compression, but also with maturation in immature layers, in cells from asthmatic patients, in cells from idiopathic pulmonary fibrosis, injury, as well as radiation. So this is an image of ZO1 showing tight junctions. This is the solid-like jam phase that I'm going to tell you about. And this is after compression, a fluid-like unjam phase. Let me go back. Jammed, unjammed, jammed, unjammed. And I'm going to show you that as the epithelial layer unjams, cell shapes become more elongated and more variable. And they do so in a very particular way that I'll tell you about. Okay. So I want to take a minute to do some history, a potted history of the unjamming transition. So it all started in airway biology in our lab. And it was this paper that kind of got the ball rolling. This was a Jabi tree pot. These are the traction forces I showed you earlier. In this paper, we have the first suggestion in the literature that cells, epithelial cells, or any kind of cell, can jam and unjam. And then it was a few years later that Jinnah Park showed unequivocally that the airway epithelium can jam and unjam. We got all the quantitative metrics, and she was also able to link unjamming to changes of cell shape. A few years later, Jen Mitchell showed the following, that we know the epithelial to mesenchymal transition is well known and is connected to cell migration. So we wondered, could it be that maybe the unjamming we're seeing is just a manifestation of EMT? It's just maybe a different face of EMT, or are they distinct? And this paper showed that at least in airway epithelium, the unjamming transition is totally distinct from the EMT. These are different phenomena. And then actually just recently, a few months ago, this paper appeared, not from our lab, but David Schwartz in Denver. He showed that in humans, that in interleukin-6 is associated with epithelial fluidization, that is unjamming in idiopathic pulmonary fibrosis. So, we don't really understand what idiopathic pulmonary fibrosis is. That's why it's idiopathic. And they showed that actually unjamming seems to play a causal role in the fibrotic remodeling of the airways. So that's an important paper. Then the question of unjamming kind of fissioned into developmental biology. Lior Adia, when he was in our group, linked unjamming to gastrulation in Drosophila. And then in other labs, O.J. Campos, who was at that time at Berkeley, he's now in Germany, showed that the long axis body elongation in the zebrafish is associated with the jamming transition. Celeste Nelson showed that in the chick embryo, the branching in the avian lung is associated with unjamming. This paper appeared in Cell from Petradou et al. Showed that rigidity percolation, which is really the same thing as jamming, is involved in phase transitions in embryogenesis. And then kind of not really under developmental biology, but Margarita DiMarzio looked at the genomic signatures of unjamming, which was kind of the first paper to do that. And then finally, unjamming seems to have found a home in cancer cell biology. This is from Peter Friedel, and they showed in 2014 that cell jamming was involved in invasion in mesenchymal tumors. Giorgio Sita in Italy linked unjamming to differentiation of cells and motility in carcinomas. And actually, interestingly, in a different paper, they made the suggestion that the jam phase is a tumor suppressive phase. And I'll come back to that. And then finally, Peter Friedel, in a relatively recent paper, showed that cell jamming is involved in breast cancer invasion. So, and now there are many more, but that got the ball rolling in these three areas. So what I haven't told you what cell jamming actually is. So let's talk about that. There are multiple different definitions of cell jamming and unjamming. A theoretician's definition is that it's a transition from a disordered fluid to a disordered solid approaching kinetic arrest where the cells stop moving. A physicist could say, well, it has to do with cell trapping, that is to say, mutual crowding or adhesion in some kind of a rugged energy landscape where the cell gets trapped in an energy well. A rheologist might say it's a solid-like regime in which cell rearrangements with its neighbor, each cell rearranging with its neighbors, are rare, intermittent, and cooperative. A microscopist, like Peter Friedel, for example, would say it's cells that are caged by surrounding cellular neighbors or ECM, extracellular matrix, where the cells become less elongated and less variable as they jam. A physiologist, like me, could say that it's a default homeostatic solid-like phase for economic tissue maintenance and mechanical stability. And finally, a biologist might say it's an energetically costly but error-tolerant episode of tissue fluidization that's associated with embryonic development, tissue remodeling, and so on. So a little more history. The whole idea of jamming was originated by Michael Cates in 1998. This is all in inert systems, not in biology. It was dramatically popularized by Andrew Liu and Sid Nagel in a Nature commentary. It was first brought into the biological literature by Javi Tripart from our lab. We wrote another paper suggesting a phase diagram that would describe unjamming. Then Jinnah's paper that I talked about already, and also this paper that I talked about already in cancer cell biology. But I want to call attention to this book. Stuart Kaufman is a well-known natural philosopher, and he's written many books. They're big, they're thick, they're complicated, they're theoretical. This one in particular, talking about what is the origin of life, or the conditions necessary for life. Now, of course, this book appeared before the discovery of cell jamming, but this book boils down to this. This is a direct quote from the book. It's kind of the final conclusion of the book. That living systems exist in the solid regime near the edge of chaos and that natural selection achieves and sustains such a poised state. Let me read that again. Living systems exist in the solid regime near the edge of chaos, and natural selection achieves and sustains such a poised state. What I want to do in the rest of the time is go from the general, this is a general statement in which he looked at many, many different systems or modeled them. I want to go to the particular and suggest the following to put a finer point on Kaufman's idea. And I would say living systems exist in the solid regime near the edge of chaos. That is, in the jammed regime, just at the very brink of unjamming, and natural selection achieves and sustains such a poised state. So that's the central idea. So that's the cell jamming hypothesis. Okay. So we decided to look at cancer cells. So these are two fellows who are in the lab, Wendy Kang and Jacopo Ferruzzi. Jacopo is now in the faculty at UT Dallas. Wendy went to Stanford. They looked at spheroids of about 1,000 cells. These are MDA, MB231 spheroids. They're a highly aggressive, invasive cancer cell. They used optical clearing and 2-photon confocal microscopy to quantify things, and they embedded these spheroids in rat tail collagen 1. And what's shown here is the behavior in two different concentrations of collagen, 2 milligrams per milliliter and 4 milligrams per milliliter. Same cells, same kind of cells, same spheres. And we were really impressed by the fact that a twofold change of collagen concentration causes a qualitatively different phenotype to arise. In the higher collagen concentration, you see these invasive tongues that invade into the collagen. But if you change the concentration only a little bit, a factor of 2, you get a totally different phenotype. You get these cell clusters and individual cells that invade. So what deeper examination showed in this paper that's cited at the bottom, that here the cells are solid-like and the cell is jammed, but the invasive tongues are liquid-like. So there must be some kind of melting, if you will, going on, going from solid to liquid. But here, again, you get a solid-like jammed core, but this is like gas-like cell scattering. So that's something that physicists call sublimation, going directly from a solid phase to a gas phase. Like in the winter, snow can disappear even when the temperature never goes below the melting point, because it goes directly from the ice phase to the gas phase. So that looks like what's happening here. So they looked a little more closely. This is finer gradations of collagen density from 1 to 4 milligrams per milliliter. And what they did is, using the multiphoton imaging, in blue, they show cells that are contiguous with the spheroid itself, with all the rest of the cells. And shown in red are cells that have escaped, cells or cell clusters that have escaped or scattered from the main spheroid. And that's again for graded concentrations of collagen. And what they found was this. So on the y-axis is the number of single cells or cell clusters per spheroid. And on the right is the collagen concentration from low collagen to high collagen. On day 0, basically nothing much happens. On day 1, a little something. Day 2, you start to see something. And actually by day 3, you see two really different behaviors with a transition at about 2.5. So as the system matures in time, this transition steepens. And so we get down here one kind of behavior, gas-like behavior, cell scattering, and up here, liquid-like invasive tongues, and a relatively sharp transition between them. So we want to understand better what's going on here. This is where Zi Chen came into the picture. Zi is a kind of a theoretical biologist, and he created a model to try to simulate. Yes?
Jeff, there is a question on the chat box. Do you want to respond to?
What is the question?
Like in the 2 milligram spheroid, is the jammed core at the cell area fraction 1?
In the jam core, it's
in close, 2 milligram spheroid.
It's no. It turns out there are cell-free regions inside the spheroid. It's described in the paper. So the answer is no, but In the shell itself, it's close to one. It's close to close packing. I think there's some small spaces between cells, but they're really small. Does that answer the question? Okay. Anyway.
Yeah.
Okay, good. Anyway, we wrote about that in the paper. So what Zichen did is he made a model, computational model, a two-dimensional model, not three-dimensional. And it's kind of an agent-based, vertex-like model, and which he had cells, which are shown here in pink, and connective tissue or collagen, which is shown here is green, as posts, obstructions. And what he included in this model is volume exclusion, cell-cell adhesion, cortical tension, cytoskeletal elasticity, viscosity, propulsion, these are propulsive particles, polarization, and ECM density. And what I'm going to show you is the results for looking at just two variables, propulsion and ECM density. So here's his model, and I'm going to play the movie. So this is at lower cell density, and you can see at lower cell density, the cells scatter. Here's slightly higher cell density. And with the increase in cell density, you suppress the scattering. Scattering doesn't happen anymore, not very much. And you get invasive tongues. And finally, at even higher collagen density, shown at the bottom, you get some expansion and not much is happening. It's looking solid-like. So what Z did is he repeated these computations for hundreds and hundreds of different conditions. And he was able to make this graph. So on the y-axis is collagen density. And on the x-axis is cell motility, the propulsive force. And I like to think of collagen density as being like a confinement pressure. Right? Is the tumor is trying to expand against the collagen creates a confinement pressure. So what you see here, and what he found was, so importantly, before I get into this, he found three different phenotypes: solid-like behavior, liquid-like behavior, and gas-like behavior, just like what we found in our experiments. So at a given degree of cell motility, as you increase the collagen density, you go from gas-like behavior to liquid-like behavior to solid-like behavior. Going the other way, cell motility, I like to think of that as being like an effective temperature. The cells are very motile, a lot of propulsive force. It's like a high temperature system, so to speak. So here, as you increase cell motility, if the cell motility is very small, right, nothing moves. But as you increase cell motility, you go from a solid-like to a liquid-like to a gas-like phase. And these lines represent coexistence lines where you can get kind of either behavior at those lines. Okay. And then down here, interestingly, he found a triple point where you can have coexistence of all three phases, and below that you go from a solid-like phase to a gas-like phase. That would be sublimation. So we had proposed in 2015 a jamming phase diagram. This is a jamming phase diagram. We proposed a different jamming phase diagram. This one is different. And Alina et al. Proposed yet a third jamming phase diagram. Again, very different from this one. But what this shows is that this puts invasion in terms of a coexistence phase diagram, and it leads to a new physical picture of invasion as being a non-equilibrium phase separation process. Now, I'm going to do a brief interlude, but I want to fix an idea that I'm going to come back to later. Okay. Has to do with evolution. So I took a page out of the book of Mark Kirschner. Literally, this is a page out of his book. He's the chairman of systems biology across the street at Harvard Medical School. And so what this is showing is the four great transitions in evolution. The first, and okay, here's a time scale from 3 billion years ago, 2 billion, 1 billion, down to the present. So the great transitions are first the appearance of prokaryotic cells, next the appearance of eukaryotic cells, then the appearance of multicellular systems, and then finally body plans. Now, interestingly, this really surprised me, but when you think about it, it makes sense. Evolutionary time is comparable to cosmic time. they play out on the same time scale. So here's the formation of the Earth about 4 billion years ago. And it turns out virtually as soon as the Earth cooled, within like 100 million years, there appeared prokaryotes, DNA, proteins, metabolism. And what Kirshner says is these inventions, these novelties were so successful that they were passed down all the way to the present, virtually unchanged. So he called that a conserved core process. Yes, appearance of cyanobacteria and the O2 production then happened. And then after that, actually after a billion years, there appeared eukaryotic cells, the appearance of nucleus, organelles, cytoskeleton, sexual reproduction. And Kirshner says that also was passed down to the present, virtually unchanged, another conserved core process. But a question is this, and this is something we've written about in years past. The prokaryotic cells have a hard wall, virtually a rigid wall. The eukaryotes have lost that rigid wall and they're soft. How soft are they? So everyone in the audience, I think, is a cell biologist. So have you ever thought if you could shrink yourself down to the size of a cell and you could poke the cell with your finger, what would it feel like? Or if you could put the cell between your fingers and squish it, what would it feel like? Have you ever thought about that? I mean, if you're a cell biologist, you should have thought about that. Well, it turns out the answer is, and we know this from the work in our lab, it has since been widely confirmed, that it turns out a eukaryotic cell, if you could poke it, would feel like toothpaste or shaving foam. That's how soft it is. And that's kind of the consistency of a cell. It's softer than the softest soft rubber by 4 orders of magnitude. And an interesting question is, why is this so? Why aren't cells stiffer, made of sterner stuff? We've written about that, and I can't talk about it today. It's a whole talk by itself. But in any event, it turns out that cells belong to the same class as colloids, suspensions, toothpaste, shaving foam. Those are all called soft glassy materials. And that behavior was passed down all the way to the present. Then there was a period of glaciation. It appeared snowball Earth, the entire Earth encrusted in ice. Then appearance of multicellularity, signaling matrix, breakup of the supercontinents, glaciation, another period of snowball Earth, and body plans and more glaciation. So the question is, where does the unjamming transition fit into this picture? I would suggest that if the unjamming transition is a real thing, it has to be kind of around here. It has to be after multicellularity appeared. So it has to be a relatively recent innovation, maybe over here somewhere towards body plans. So I'm going to come back to that. So a deeper dive into cell jamming. Okay. How does unjamming actually work? It turns out, this is Stuart A. He came to our lab, I think, in 2015, and he took a novel and radical approach to this question. He said, much to everyone's shock, he said, think about a bucket of sand at the beach. And we said, well, what does that got to do with cells? He says, it's got to do with it's everything. He said, you can, in the beach, you can stand on the sand, you don't sink into it, but sand can also flow. You can tip the bucket over, you can, shows here the quarry. Sand can flow or be solid. And he said, same way with epithelium. It can be solid-like, or I showed you, it can flow like a liquid. And he said, further, both systems have some deep connections. They're both collective systems made of individual subunits. They're both close packed. They're both disordered systems. They're not ordered, they're not crystalline, and they're both jammed. Jamming occurs in both systems. They both exhibit fluid-like and solid-like phases. And it turns out the number of packing configurations is huge. I read somewhere, and don't hold me to this, but if you counted the number of different ways you can arrange the sand grains in a bucket of sand, it's more than the number of electrons in the universe. So there's a huge number of possible configurations, but so too for epithelium and the way epithelial cells can pack. And then finally, that the basic physics has nothing to do with thermal agitation, KT, right? Brownian motion. If you look at the sand grains in a bucket, they're not moving. You don't see Brownian motion because these particles are too big. KT is not enough energy to move a sand grain. And similarly, KT, thermal energy, is not enough to move a cell. You can get millions of myosin motors can move a cell, and cells do move, and sand grains do move, but it's got nothing to do with KT. So this is athermal systems that are not at thermodynamic equilibrium. So then some statistical physics happens, which you can read about in this paper, and it led to this hypothesis that as the epithelial layer unjams, cell shapes become more elongated and more variable and in a very particular way. So again, I showed you this before. That's jammed, unjammed, jammed, unjammed. And it turns out if you look at the aspect ratio of cells, they follow a particular distribution. It's called a K gamma distribution, and it's predicted by the theory. So it fits well in many systems that I don't have time to talk about today. So finally, the last topic. And this is the main topic that I wanted to get to. Where does physiology happen? Well, what do I mean by this? So here's the phase diagram we talked about before, and it has experimental support. So this is a phase space, right? You can imagine cellular systems anywhere in this grid. And the question is, are all these loci equally conducive to complex cooperative behaviors that are typical of life or evolution? Would you expect that some regions would be more conducive to life than others, more conducive to development than others? So in this model that Z Chen put together, all the cells were identical to all the others. So there was no heterogeneity, but of course we know that in any real system, the cells are going to have different properties or some distribution of properties. So let's do a Gedanken experiment, a thought experiment, and let's consider a heterogeneous system where the cells are not all alike. Now, just an important caveat. Some of you might not like this phase diagram, and that's fine. It turns out the argument I'm going to give postulates that there exists a phase diagram, but its particularities don't matter. So if you don't like this phase diagram, the argument I'm about to make would apply to any phase diagram, assuming that one exists. So here's the phase diagram. Again, solid-like, liquid-like, and gas-like phases. Let's imagine that there are let's put ourselves deep in the solid-like regime, frozen, just frozen. Cells are just jammed like mad. But there might be some cells that represent kind of a melted pool that are in a frozen sea, kind of like this. So we get a melted pool isolated in a frozen sea. Now, if there's any complex behavior that happens in this pool, like a fish pops up or a duck jumps into it, those behaviors don't propagate because they're shielded by the ice around it. So you can get a local perturbation, but that only causes a small global change. And this might fit with, again, what Giorgio Schita said, that jamming is tumor suppressive, right? If this were a tumor, that tumor would have a hard time invading. Now, on the other hand, let's consider that we're deep in the melted regime, chaotic like mad, a lot of motion going on. This is kind of like an iceberg in a melted sea. So you can get cooperative behaviors, right? This iceberg could calve, could split, but it's not going to propagate because those perturbations are going to fail to percolate across the system. They're just going to get dissipated in the fluid. So this would be a tumor permissive system. So the thing I want to highlight here is on the left, we get complex behaviors, but they're shielded and therefore they can't propagate. Whereas over here, we can get cooperative behaviors like calving, but they get dissipated and they can't propagate across the system. So where can you get in this phase diagram a region where you can get complex behaviors that are also cooperative? Well, there's only one place, and that's right at this jamming phase diagram. So I would argue that the unjamming phase boundary behavior, you can get both behavior that's complex yet cooperative, right at that boundary. And I would say that's where physiology happens: remodeling, wound repair, development, branching, growth. That region is conducive to all of those things. They're also conducive to certain pathologies like asthma, idiopathic pulmonary fibrosis, invasion, cell scattering. Now, phone is buzzing. Okay, so I kind of know what you're thinking. You know, is this idea plausible, or is it nuts? Well, I don't know, but here's some recent data. This is from Joseph Case's lab in Leipzig, a paper that's in press, and he took explants. I think from 1,300 women. I think he said these are explants of mammary tumors. And they took AFM and they scanned, this is from 0 to 500 microns, and they scanned to measure the stiffness and the stiffness heterogeneity within these explants. And here, blue means soft, red means stiff, 0 to 2,500 pascals. So here you're seeing a melted pool, if you will, isolated in a frozen sea, kind of like what I've shown above. Now here's explants from a cervical tumor. Same kind of thing, but here you're having a stiff region. So here you're finding a frozen iceberg isolated in a melted sea. So there's some evidence that maybe these kinds of ideas had applicability, at least in cancer biology. So coming back to Stuart Kaufman, living systems exist in the solid regime near the edge of chaos. That is to say, in the jammed regime, just at the brink of unjamming, and that natural selection achieves and sustains this poised state. Okay. Now, to wrap, I've got 2 or 3 slides just to wrap up, and then I'll be finished. I'm glad to see I'm on time. So shown on the left is a jammed system. This, if you will, is a poised state. It's kind of ready to unjam. It's ready to do a whole bunch of functions, but it's not. It's poised. It's jammed. It's solid-like. It's not migratory, and it's stuck or frozen. Here on the right is an unjammed phase, if you will, an excited phase. It's unjammed, fluid-like. The cell collective is malleable, so it can reshape, migratory, and it's unstuck or melted. So this is the jamming hypothesis kind of in different words, and that is that natural selection favors life near the brink of cell unjamming. And the corollary one is that physiological functions arise from chaos, not from order. Right? When we think of cellular systems, you do see ordered systems, like in the eye of a fly. But in most human systems, physiologic functions arise from chaos. And the chaos is, that is to say, the unjamming condition drives. It's beneficial because it can drive exploration of all possible configuration spaces. We know from data that during development, cells unjam. We know that during maturation, they progressively jam. So jamming they're both physiologic functions. In cancer cell invasion and pathology, cells seem to unjam. In mechanotransduction, they tend to unjam. And in asthma, idiopathic pulmonary fibrosis, RV infection, injury, radiation, they tend to unjam. So where does this leave us? I'm getting near the end. Here's the molecular phenotype of a cell. We know that genes are very important in terms of determining the molecular phenotype. No news there. And we know that there's a microenvironment. And the microenvironment can include growth factors, inflammatory factors, stiffness of the matrix, mechanical compression, infection, injury, radiation, and so on. And they can also affect the molecular phenotype. And then the molecular phenotype would determine the biophysical phenotype at the single cell level. And then there can be mechanical events that actually feed back and change the molecular phenotype. And then there's also mechanogenomics. We now know that mechanical forces can change gene expression. Okay, That's a single cell level. What about tissues, multicellular systems? We can get solid-like behavior, poised, stable, stationary, economical, or fluid-like behavior, malleable, migratory, or expensive. And the transition between these is either the unjamming transition or the jamming, or the jamming transition going the other way. And then that would then the unjammed system, fluid-like, would be permissive of development, remodeling, repair, fibrosis, and invasion. And then finally, there's natural selection, which would apply to then feedback on how genes evolve and are expressed. So interestingly, the time scales are divergent. We know the level of signaling, that's very fast. There are calcium sparks, channels, diffusion over very short distances is very fast. Less fast is transcription and translation, to some degree, protein folding. Unjamming transition or the jamming transition is pretty slow, hours, multiple hours or days, whereas natural selection is glacial. So the notion is that we have divergent timescales, and the question is, what physical conditions started this ball rolling? And I think it has to do with extreme cellular malleability, having to do with unjamming, acting over cosmic timescales. And that puts the unjamming transition here. And also there's a big role for the mechanical properties of individual cells. So that's it. I'm going to stop there. I want to thank the people who contributed to this work, especially Chan Young Park, who was our laboratory manager, Jinnah Park, who did a lot of these experiments I described, Jim Butler. Nothing good happened in our lab, ever happened in our lab without Jim's participation. Jennifer Mitchell played a key role. David Waits played another key role. Jacobo Ferruzzi, I mentioned. Ming Guo at MIT, I didn't mention. Zichen, I mentioned. And then Daping B, Max B, and Herb Levine at Northeastern University. So I'll stop there and take questions.
Thanks, Jeff. You have a good news. There is no audience left while you are speaking.
Good. Okay. So, questions, comments, criticism?
Chris has a question.
Stunned silence.
I didn't know if first we're going to open it up to trainees.
Oh yeah, right. Yeah, please. Trainees first. Thank you, Chris. Okay, sounds like no trainees.
Charlene has, I think, a comment. Can you just unmute yourself and go ahead, please?
Can you hear me?
Yes.
Okay, great. Thanks for the talk, Jeff. I know that there's been a desire to have people to participate more, so I will do my best here. So I'm going to say when I gave my talk last year, people didn't really interact with me, and I think that the reason for that is that these are pretty different fields. So I'm an epidemiologist, so
right.
The person who did interact with me suggested I do bench work, and so to be honest, if I went to a conference, I wouldn't attend cell biology talks, not because they aren't interesting, but because most of what is said isn't quite relevant without a collaboration. Mm-hmm. So, and I know it's the same, it goes the other way too. So cell biologists wouldn't usually attend a talk on Mendelian randomization either, by choice, right? They wouldn't, because you can do other things. So for the betterment of my learning, while you were talking, I particularly appreciated when you had the author names on the slide, because I could look them up right then. The Gerhardt and Kirschner papers, the 1998 2007 papers on the evolutionary transitions. That slide was fascinating. I perked up, I got excited, my heart started racing. So I pulled both of those papers just then, and just reading the abstracts was great. I couldn't find that lovely slide. I don't know if that was from you or in a different paper or whatnot. But
it's in Kirshner's book. Here's a book.
Oh, it's in the book. Okay, yeah.
And if you send me an email, I'll send you the reference.
That's great. That would be lovely. And so thank you for the talk. And that's my main participation.
Well, thank you for the
You're welcome.
comment.
We have a question from Hiren. Can you please unmute yourself and ask a question?
Yes, of course. Sorry. Yeah, I read your paper where you showed that the cell aspect ratios are or the rescaled aspect ratios of the cells are K-gamma distributed,
Right.
and this is like this distribution was found for the equilibrium volume packings in granular systems. And I would like to hear what or why do you think those aspect ratios have the same distributions?
As in granular systems.
Yeah.
Yeah. Okay. Yeah. We explain that in the ADIA paper, but the idea is this. I have a slide that describes it, but I'll try to do it just in words. We understand in kind of normal molecular systems that KT, thermal energy and collisions, are what allows the system to interact with, allows particles to interact with its neighbors. But we know that in granular systems, KT plays virtually no role. So it was, I'm blanking on his name. It will come to me. That's the sin. Wait, let me actually find the slide and put it up here. Bear with me. Share screen. I can't find. I'm not going to try to find it. But the idea is this: that in granular systems, volume seems to take the place of temperature. So the thought is that the volume gives particles space to rearrange. Now, in free volume around each sand grain, for example. Now, in cellular systems that are confluent, there is no free space, or very, very little. So the thought is that it has to do with cell shape. That's how cells can rearrange, by changing their cell shapes. So in each case, whether it's thermal systems, granular systems, or cellular system, it turns out that it's the microconfigurations of the systems that controls the physics. And it turns out by analogy, or even by analysis, the K-gamma distribution that's found in granular systems, it turns out carries over to cellular systems, because it all has to do with microconfigurations. Sorry, I couldn't find the slide, and I'm blanking on the.
No problem. But, thank you very much for the answer. So, yeah, the analogy is just degrees of freedom is volume in granular systems, and degrees of freedom is the elongatedness of the cells in confluent cell systems. Okay, thanks.
Mm-hmm.
Okay, we have the hand raised by Sherry. I think that you are our trainee. Can you please unmute yourself and go ahead?
Yeah, thank you. My first question is that why exactly does being near the brink of cell jamming gives a life evolutionary advantage? Like, why this?
It's because if you're deep in the frozen regime, it's hard to do anything, because you can't transmit information over bigger distances. Whereas if you're in the fluid regime, it's totally chaotic. But if you're right near the jamming transition, you can have the best of both worlds. You can have cooperativity plus malleability, so that you can do things. So that's the hypothesis. And all I did was really put a finer point on what was done before by kind of blanking on the name of the book I just showed. But you get cooperativity plus the ability to transmit over bigger distances, because it's solid-like. Oh, by the way, the name before was Edwards, the Edwards conjecture. is the one for granule material. I forgot that.
Thank you. And my second question is, do you think there's a reason that evolutionary time is comparable to cosmic time? Like for example, the climate change on Earth could driven the evolution.
I'm sorry, I don't understand the question. Could you repeat?
Like, is there a reason that the evolutionary time is comparable to cosmic time? Like,
Oh, a
for
reason.
example, yeah, a reason, yeah.
No, I guess that's what we have. I mean, it's just the fact that prokaryotes appeared kind of immediately as soon as the Earth cooled, and but then for reasons that are not altogether clear, it then took 1 billion years for the first eukaryote to appear, and 1 billion years is definitely cosmic time. And why did it take so long? Well, I think no one actually knows. And I know that there's an author called Cavalier Smith who's written a lot about this. And based on genomic analysis, they believe that the transition from prokaryote to eukaryote happened only in one place at one time, in one cell. So there was nothing inevitable about eukaryotes appearing. It could have been that our planet evolved to be prokaryotes only. There was nothing inevitable about eukaryotes. So instead of appearing 1 billion years after prokaryotes, it might have been 10 billion, in which case we wouldn't have any eukaryotes. So that's contingency in biology. So it turns out these accidents that happen, they play out over huge timescales, that are comparable to cosmic.
I see. Oh, that's very intriguing. Thank you.
Okay, due to the interest of time, we will have a last question from Professor Stuart A. Thanks for your patience. Go ahead.
All right, I get a question. Can I do 2 questions? Because it's kind of 2. Wonderful talk as usual, Jeff. Always beautiful to learn about the cosmos as well as molecular biology in the same talk. I was curious about 2 things. One of them being, obviously the implications of some of the work that you're doing in cancer would be to restrict the migration of cancer cells away from the primary tumor and thus limit metastases. Are there ways that we could be potentially modulating the jamming transition inside of a tumor to then be more restrictive to that? And then another question, well, you can, go ahead and answer that one real quick, and then I'll give you another really fast.
The answer is probably yes, but I'm not the guy to answer that question because I don't know enough about cell signaling and those sorts of things, but other people do. And the answer is probably yes. And in terms of the practical side in cancer, I know Joseph Case was on the call. I don't know if he's still here. He has a paper showing that by looking at biopsies from women, you can predict whether or not they're going to go on to metastasize. And it turns out these predictions are more accurate than lymph node status. So, so the suggestion is by looking at a static slide, is because we think shape is related to migration. You just look at shape and you do the statistics with some machine learning, you can predict which women are likely to metastasize and therefore need aggressive treatment and which ones don't. And it does better than lymph node status. So that's a very practical thing. So that paper is in review, but it's a practical outcome of the idea of cell jamming.
Yeah, that's amazing. And one really quick final thought was that you made the case that the jamming transition would be affecting the propensity of cells to migrate away and metastasize. I wonder whether their ability to settle in a target tissue would then also be affected by the jamming transition in that tissue, and how accepting it is to the different growth states and migration states of the metastasizing cell. We know that different tumors tend to metastasize to different organs, and so a lot of that is thought to be based on blood and lymph and other things, but maybe jamming is also an important.
I think so, because it used to be thought for a long time that metastasis has to do with a single cell escaping from the primary tumor and seeding somewhere else. And even most of the cells don't make it, but it's a single cell phenomenon. Recent data shows that it's not. It's a collective cellular phenomenon. And Andrew Ewald showed, I think a year or two ago in a paper that appeared, I think in Nature, that E-cadherin is necessary, this is in mice, is necessary for metastasis. So that suggests that these cells are somehow collective, and when they get to the seeded locus, finally, they probably rejam. And the jamming may have a lot to do with it. That's what stabilizes the tumor. So could be. We don't know. We don't know, but that it definitely, I think it's a reasonable hypothesis.
Thanks.
I see there's one more hand up.
Yeah, it could be a very burning question. Bobby, do you want to unmute
Yes,
yourself?
can you hear me?
Yes.
Sure. Yeah, we can.
Yeah, thank you, Jeff. This is a quick question. I think it's real quick. Just in the case of jamming and unjamming transition, you said it requires some compression, like just for anything for jamming, unjamming, it requires compression.
Oh no, it doesn't. No, it doesn't require compression at all. Compression is one of the stimuli that will do it, but there are lots of stimuli that will do it without mechanical compression. So maybe I
Okay.
wasn't on that.
No, that's fine. What's just one example, I guess, of another stimuli as opposed to compression? Like, how does it, instead of compression, it would be.
Yeah, but stretching will unjam. We didn't publish that, I don't think, but we know stretching will unjam the system. Poking it in different ways will unjam it. Chemical stimuli can unjam it.
Okay.
Radiation, ionizing radiation will unjam it. So there are lots of pathways to unjamming, lots of stimuli.
Okay, thank you.
Yeah, thanks for the question. Okay. Well,
Okay,
then,
great. Yeah, thanks for the splendid talk, Jeff, and it was very inspiring as always, and people learn a lot from your talk. The next week, also the audience, thanks for your questions and participation. Next week, we will have in-person work in progress seminar in room 1302, but also will be available through the Zoom call. But MIPS trainees and faculty staff are encouraged to attend in-person seminar in room 1302. And it will be by Dr. Nora Bankova from the Brigham and Women's Hospital. Thanks, everyone.